Hi, Daniel.
Good morning, Professor.
Morning. How are
How
you
are
doing?
you? Oh, I'm
Oh,
doing well. Sorry.
no problem. Hopefully, Zoom has been kind of slow. Hopefully, with a small group, it won't be too bad today.
Yeah. I
What?
was I was rushing to try to find the Zoom link because I didn't know where it was.
Oh, are you taking other classes?
Yeah, I'm taking the Chem 130 class.
Oh, how's that going?
It's not too bad. It's kind of very similar to how this course is with lecture because I also have a lab with that class.
Yeah, I've taught
Um,
130. I like that class.
Yeah, it's not too shabby. It's just
And Dr. Bowers
basics.
is great.
Oh, I actually have it with Dr. Edward Alexander from
Oh,
Mesa.
oh, okay.
Yeah.
Which book are you using? Are
It's like a customized one.
okay.
Uh, Let me see if I can find it real quick.
There's probably some overlap on the content, huh? Hmm.
We're using a custom Pearson's one.
Oh, Timberlake, probably. Is it
Uh,
Timberlake?
I'm not too sure. It's
Is
like
that
custom made for Mesa College.
because Timberlake is Karen Timberlake is the author of the book that does 100 and 130 for Miramar. I don't know if it's the same book. Oh,
Karen. Oh, yeah, yeah, it is. It is.
yeah.
Karen
I like
Timberlake.
that book. I think she does it's at a good level. I think it's more readable than our book.
Yeah, it's more laid out, I guess.
Mhm.
Um there's a lot more kind of formulas in it than what we're doing.
Formulas.
Well, not really formulas, but kind of rules, I guess I would say. Like I don't know. This textbook is just laid out differently than our textbook.
Yeah. Well, ours, I feel like ours rambles a bit. It's hard to pick an appropriate text for this class
Mhm.
because it's not a biochemistry class headed towards a chemistry major.
Mhm.
Um, so I think I don't know. I don't know that there's a better option out there, but I don't love this book.
Uh,
So you see my PowerPoint?
yes, I do.
Okay. Where is everybody? What this group? Everyone's doing well. Um, there's Dustin. Um, like I was pretty happy with the test.
Mhm.
I thought I don't know, I felt like the questions hit what I wanted people to hit, and from reading the answers I feel like people understood the important parts that I was looking
Mm.
for.
When do you think you'll have the grades up for the exam?
Oh, I posted them, didn't I?
Oh, did you? I didn't see a grade yet.
Oh, I'll look.
Okay.
Yeah, I graded them pretty fast. Um, and yeah, it might be a setting on Canvas. I'll have to look.
Oh, okay.
Yeah,
They're on Canvas.
they are. Okay.
Yeah.
Hi, Dustin.
Hi.
I don't see myself on camera. Do you guys not see me?
No.
No.
I don't know it's probably because I have my document camera on, but that's weird. I don't know because I have my video on. Huh. Oh well. Do you now see the PowerPoint?
Yes.
Okay. How's the homework going?
I just kind of started yesterday, actually.
Okay.
Yeah.
Same.
So I'm not too far in.
Oh, okay. I probably should have made this one due sooner, but I felt bad after the exam.
Mm.
Although that feels like it was a while ago now. Um, I wanted to get a critical mass for this because I want to introduce the idea of the project. We have a 50-point project that we have to do. And I thought right now, I mean, generally it's open to whatever topic you want in biochemistry, but I thought right now it would be really nice and maybe more interesting to do something focused on coronavirus.
Yeah.
Um, yeah, there's so many questions we have now, right? And um, it's it's relevant to us personally, but also a lot of the things that are being discussed in the news are related to things that we've learned or are about to learn. Because next chapter is on viruses. It doesn't go in very much depth, but I have colleagues over at USD that they work together, like they combine resources with faculty at other universities, and they put together an activity on COVID, and I wanted to show you. It's pretty rich. The I got to do a different screen share. The amount of material they've amassed is pretty impressive. Um, so there's like videos that have been vetted. So now you see this, right? The project.
Yes.
Okay. So the idea is that you guys pick a topic for next Thursday. We have to get moving on it so that you aren't totally crushed at the end. So pick a topic for next Thursday and include 2 or 3 references. And I have there's another file that has a list of a ton of references. So you could go through those and see what is interesting. And the project submission could be either a 5-page paper or a 5-slide PowerPoint presentation. And because I think it would be nice to get together here and just talk about what we learned. So either one would be fine. And it's kind of funny. Dr. Bauer did this last semester, and she had a day where people could do posters in class, and she was sort of surprised how many people chose to write the paper rather than talk in front of their peers. For me, I would rather talk, but but I'm used to that now. And when I was your age, I probably would have picked the paper. um, but I think it would be nice to compile a comprehensive repository of information about the virus. And some of the questions that I was thinking, I'm gonna let Michelle in. She just is in the waiting room. Hi, Michelle. You can't see me. I don't know why. Or is she still connecting? Hi, Michelle. Sorry, I don't know what's going on with my video today, and I don't want to mess with it because yesterday I had to I had like the Zoom session from, I don't want to say the word, yesterday. I had to reboot, and then when I rebooted, my document camera wasn't working and it was really slow. So I'm gonna go with what's working. So I'm just talking about, we have a project in this class, and I'm suggesting that we focus on the biochemistry, epidemiology, and therapeutics of COVID-19. And I thought that would be interesting. I know you're dealing with it on a daily basis, and Dustin probably is too. So some of the questions are like talking about the antibody test. What does it demonstrate? What does it not demonstrate? Right? We know it doesn't tell you if you have active virus. And then will there be cross reactivity with other tests, right? Because there are other coronaviruses that are not as pathogenic as this one. And I know when we went to Costa Rica, my daughter got really ill and we thought she had dengue fever. And when we came home, we did the antibody test to see if she had dengue. And she tested positive, but they said you don't really know if she had it unless you track the titer, because it cross reacts with the vaccine. I think it was for yellow fever. So, right. So is there going to be cross reactivity? Like if I have a common cold that happened to be a coronavirus, is that going to test positive for the antibody for COVID-19? So that's a challenge. Another one is, the vaccine. So there's never been a successful vaccine developed against a coronavirus. So I would like to know how are vaccines made, what progress do we have now, and what do we think the likelihood is that, that this is going to work. There's also the coronavirus is related to MERS and SARS. So questions about that. Some of these are a bit more biochemistry oriented, but describe the genomic and protein virion structure of coronavirus. There's actually really good references in that other document compiled about this. And I found a video last week that was a really good presentation about the mechanism of infection, how the virus invades cells. And I'm looking to find it again. I haven't, there's others, but this one was really at an appropriate level for us, I thought. So I'm going to try to find that. Substrains of COVID, are there substrains of COVID-19 virus, right? So right now there's some people have suggested that New York has it so bad because they got a different strain of the virus than what we've gotten on the West Coast. I doubt that it's changed that much. I think it's more about the living circumstances. Let's see. There's more, like number 15 down here. What test is used to detect COVID-19, right? So in patients. And so again, this is, are we doing an antibody test from blood or a PCR test from nasal swabs? And one of the things about the PCR test is it detects fragments of virus, but it doesn't tell you whether or not it has the ability to infect, right? So it would be interesting to look at different kinds of tests and how they work. And then the SARS invades by binding ACE2. That's what that video was talking about. It was really good. So, and so understanding the mechanism of infection also gives you some insights on how to find a therapeutic for it. Down here, number 16, what's the pathology of the virus? What causes the pneumonia-like symptoms? Some viruses are lethal as they rapidly mutate. So how mutable is this compared to other viruses? So you get the idea. There's really a lot of questions that we could have in the project, and I think I would find it really interesting. And I also think things are changing so fast. It would be really neat to see what we come up with now. And then you guys will have moved on, but I would reflect back later and see how it turned out, right? Because we're like in the middle of history that's unfolding, and to do some research now would be interesting. So what I was saying is, I would like people to pick their topic, like pick a question or two to address, and email that to me. I don't want to have too many people working on exactly the same thing. I don't mind if there's 2 people doing similar topics, or even if they want to work together, that would be fine with me as well. But I don't want to have 8 things, 8 projects on the same question. So I just want to have you email me the topic and the question, the references that you're going to use, more so that I can make sure it's an appropriate scope. So what do you guys think? Dead silence. Let me unmute.
I think I'm like fine with the idea.
Let me see. Oh, I'm trying to find the resources. So I put a folder inside our homework and activities folder on Dropbox, and there's a list of references. I'm trying to find, there's one Word file that has links to a lot of stuff. I'm trying to figure out which, and what I'll do is I think it's this one. Yeah. Let me do a different screen share. Here it is. Okay. So you guys see that where it says resources now?
Mm-hmm.
Okay, so like this one is really good. It's so funny. It's a video, and she basically says she's a board professor at home, and it's all hand drawn, but it's really, really good. And so there's a few YouTube videos, and then some reviews and some websites. Oh yeah, and then some of the one of the topics was, pick a potential therapeutic and talk about how it's supposed to work and what the likelihood of it working is, right? I think people are less bullish on chloroquine than they were before. But again, everything's changing so quickly. Here's one about remdesivir, because I think that has shown some promise. So these are all potential references. We haven't done as much of this, but these are protein sequence alignments. This is the kind of biochemistry I spent my life doing. But you can see how similar these are amino acid sequence of the E protein from MERS, SARS, and COVID, and you can see how similar they are and where the differences are. That's probably not as relevant or as interesting to you guys. So if someone really wants to do a project on some other biochemistry topic, I'm okay with that. I just think it would be nice to compile a lot of information about this right now, because I like to be able to tell my friends why they should wear masks. Okay, let me go back to And what I will do is, I will post links to those specific files on Canvas, and I'll create the assignment too. I think, yeah, that way, because then you have it all in the same place. And you can just submit your topic, I think, via Canvas. Then it's all in one place. Chapter 14 homework is, I have it on the calendar as due 5/6, which is not that long after 5/4, but we've had a bit of time to do 13. Let me know if you're feeling too stressed about getting chapter 14 done. I haven't typed that one up yet. I'm hoping to be able to get to that maybe tomorrow. I have to tell you, this going online, I feel simultaneously like I'm letting down students, but I'm also constantly behind. I'm constantly turning the wheel to get stuff out and having to pick what gets done and what I have to do differently. I've been trying to write an exam for my general chemistry class, and I'm an embarrassing number of hours of labor into writing the exam because I'm trying to do it all on Canvas. It's like 6 or 7 hours of writing it so far. So I guess you guys don't have questions on the 13 homework because you haven't gotten that far along in it.
I don't have any.
Okay. All right. Well, do you guys know how PCR works?
Yeah.
Trying to get the gist.
Okay, let me, I've been going through the homework. Some of it, it seems pretty, like the chapter, I think like it has been a lot along the way. The chapter is kind of overwhelming, but when you go through and read the questions and answer them, it's not so bad. Okay, so I just did a screen share. Do you guys see polymerase chain reaction?
Yes.
Okay, let me close that. Okay, so and one of the reasons why I wanted to talk about PCR is because that's how the virus is being detected, and that stands for polymerase chain reaction. And what makes it so valuable is that it's this method where you can take a really, really small amount of DNA and make massive numbers of copies of it in a short period of time. Oops, that's not what I wanted to do. I was trying to highlight. And you can do it without having to isolate without having to isolate the DNA. And the other thing is, so you can repeat this. You get some DNA sequences, and you have primers that you get complementary pieces of DNA that allow you to direct what gets copied. And so a copy of DNA is made, and then the DNA is melted, and another copy can be made. And you can do all of this without having to isolate the DNA, which saves on steps, and you can start with a very small amount.
Don't you need to like at least know a bit of it to recognize what primer to use?
Yes, yeah, you do. And the primer design is actually the important part, because that's the part that can fail.
Yeah, because you need it to bind at the beginning and also not bind with itself.
Yeah, so you pick primers that are, can you guys, is this a big enough image for you guys to see? Or should I, let me see if I can make this bigger. No.
Oh,
Oh,
yeah, that works.
That worked.
that works. Okay. Let me go to. Yeah. So the primers. So here's the targeted piece of DNA, right? And it's duplex. So you have to melt it. You have to raise the temperature so that the strands separate into single strands. And you pick primers that are on either ends, opposite ends of the two strands. The primers have to be able to bind the template that you're interested in. You also don't want them to bind to each other, right? If the two primers are complementary, they're going to bind to each other and not to the target DNA. The other thing that can happen is sometimes you could have a sequence on the primer that has secondary structure, right? If the primer forms a hairpin with itself, that's going to prevent it from forming to forming the duplex with the targeted sequence. And then the other thing is, so you load the primers and then you give it Taq polymerase. That's Taq stands for Thermus aquaticus. That's an organism that grows in the geothermal vents. And so it's very, very heat stable. And so it shows you can do, they're doing the polymerase at 72 degrees Celsius. That's very hot. And most proteins would fall apart at 72 degrees Celsius. And so that's why Taq polymerase is so valuable, is that you can raise the temperature high enough that the DNA that you're targeting is single stranded and then do the sequence. If most enzymes would fall apart, and you'd have to lower the temperature to do the polymerase reaction, but the problem then is that you have more duplex DNA than you want instead of the single stranded. Does that make sense?
Yes.
Okay, so then polymerase is done, and then you have to raise the temperature again to separate. So you go from 1 to 2, and then from 2 to 4. So you again separate strands and anneal with the primer, and then do the polymerase reaction where you synthesize the DNA. So it's powers of 2, right? 2, 4, and then you get 16 and then 32. So it very rapidly, with each temperature cycle, you get a doubling of the quantity of the interest of DNA, and you do it such that it winds up swamping out everything else that's in the mixture initially, because there could be other things in there initially, but these primers allow us to select out the parts that we want. And so one of the so this is the method that's being used to detect the virus from nasal swabs. And I wanted, one of the things that I wanted to bring up was, I think I told you the story about Kerry Mullis. He's the guy who came up with this. He came up with the machine that cycles the temperature. He wasn't the first person to isolate Taq polymerase, and he wasn't even the first person to use this idea of temperature cycling to make copies of DNA, but he made this machine that cycled the temperature. And for that, he won a Nobel Prize. And the company that he was working for attempted to patent Taq polymerase. And think about, and I think initially I always forget, I looked through I think initially the patent was granted and then it was reversed. But think about what's going on now and the limitation for reagents for the PCR test for detecting the virus. Imagine if there was just one company that had total control over the production of Taq polymerase, and the entire world is dependent on that enzyme to be able to diagnose the virus. So it's another consideration that I hadn't thought of until now, like, about the consequences of awarding patents to biochemical things. I know my friend, she works in a a facility in Montreal and at McGill, and Montreal's got, it's got a concentrated population, and a lot of people tend to go to New York on the weekends there. So they've got a pretty, they have a fair amount of virus moving quickly, and also a lot public transportation is very popular there, so it's harder to prevent transmission. And she said that the labs in the university were giving their Taq polymerase to the clinical groups to help with the virus tests. So it's sort of interesting to think about that. I was sort of surprised that we haven't heard more about the Taq polymerase supply in terms of reagents, but maybe that's too much detail for the news.
It's not important enough.
Yeah. Okay. So in terms of that, everything else I was looking through, it's probably worth talking about plasmids. Okay. So there's a lot about expression vectors, and so an expression vector, let me see if I have my pen, I can write on the screen. So when you clone a protein and you're using bacteria to make protein, what you do is you let me see, I think this is a pretty good one. So when you're trying to make protein and you in the lab, and it's a cloned protein, you're generally using the machinery of, most of the time it's E. coli, to produce the protein of interest. And so you have some synthetic gene, and what you do is insert it into what's called a plasmid. Plasmids are double-stranded circular pieces of DNA that naturally occur in bacteria. They're not part of the genome. They come and go with bacteria, and they often give bacteria resistance to things in nature. But what we do is we have taken one naturally occurring and modified it for our purposes. And so these plasmids, you insert the gene that you're interested in. This is about 5,000 base pairs. I forget what the E. coli genome is, but I don't know, it might be like 1 million. So this is a small piece of DNA, and you transform E. coli. That means you get it inside the E. coli cell. And that takes like an hour or two. And it has roughly like a 1 in 10,000 efficiency. So maybe, if I remember correctly, I used to do this a lot, but like 1 in every 10,000 of the plasmid will get in, but we have a way of selecting the E. coli to get the ones that only take up the plasmid. So here's my E. coli, and we add an inducer, just like in the transcription factors, when allolactose turned on transcription of the lac operon. You guys remember?
Mm-hmm.
So there's in the lab, it's almost always, it's a allolactose analog called IPTG, and you turn on the lac operon inside E. coli, and you tell it to turn on transcription, and we've put our protein, our gene of interest, in that operon, and so the E. coli cell makes tons and tons of the protein, and you can trick the E. coli so that the protein that you are trying to study winds up being like 70% of the total protein in the bacteria. It's the major protein component. And when you are trying to purify something and it's 7 out of every 10 molecule, it's easy to purify. When it's 1 in 1 million, it's much harder to find, you can imagine. And so you break open the E. coli cells and then you purify. So that is a very, very widely used process in labs, because almost all proteins that are studied these days, they're almost never isolated anymore from a natural source. They're virtually always cloned if possible, because then you can make changes to them and study how the changes impact the protein. And so the plasmid is this part here. And the plasmids, this is this double-stranded piece of DNA. And let's see if I have a, what's the, I think this is the one I want. Yeah, so the plasmid, see, it's about, this one's about 4,000 base pairs. It's this double-stranded circular piece of DNA, and they're all mapped out. These abbreviations in italics here, those are all sites for restriction enzymes. And those are enzymes that cut DNA in very specific sequences. And so the plasmid will always have ORI-C. This is the origin of replication. If this double-stranded piece of DNA doesn't have the origin of replication, once it gets inside the E. coli, it's not going to be replicated, right? We know that DNA replication begins at an origin of replication, and in E. coli, it's ORI-C. So this has to be a part of the plasmid. The other thing is there has to be, usually there's 1 or 2 genes for resistance. This AMP-R, that is the gene for ampicillin resistance. So when this plasmid gets put inside the bacteria, that bacterium can live in the presence of an antibiotic. Does that make, so this makes beta-lactamase, which breaks down penicillin and ampicillin. Generally, E. coli, they are killed in the presence of ampicillin and penicillin, right? That's why we use antibiotics, to kill bacteria. If you put this plasmid inside E. coli, then you can put in the medium ampicillin, and then all the E. colis that don't have this plasmid will die, but the ones that do will survive. And so that's one way of selecting for our transformation process. So there has to be, and we generally use ampicillin. And then there's also what's called a multiple cloning site. They're showing, in this one, they're showing using the restriction enzymes. So the restriction enzymes cut, doesn't look optimal actually, because it's not a sticky end. But the HIND3 and BAMH1, those are both restriction enzymes. Oh, show here. And see how this is what's called a sticky end. There's an overhang of bases on either end. And see how they're different? So if this is my gene of interest that I'm trying to insert into this plasmid, having sticky ends and we want to make the ends on the plasmid complementary, that forces our gene of interest to go in one location and in one orientation, right? It's gonna be the correct sequence to code protein in one direction, and in the reverse direction, it will code nonsense. Does that make sense? Mm-hmm. Questions? So the sticky ends, if they're, I can't find my, oh, here it is. It was stuck on the, okay, so blunt ends would just be if both pieces ended like that. And then this could go in here. It could go in this direction, or it could go in that direction. And only in one orientation does the gene code the protein that we want, our gene of interest. So what we need is our plasmid has to have restriction sites, and then the area where this goes in is called the multiple cloning site, MCS. There'll be there's a bunch of different restriction sites that you can pick different restriction enzymes to put your gene of interest in, but that's a necessary component. And this multiple cloning site is going to be downstream of a transcription start, so that when we turn the operon on, our protein will be made. I'm trying to think of terms that are a little bit tricky if you haven't used them before. Oops, let me go back to that. Have you heard of, are you guys familiar with the blue-white screening? Dustin, have you heard of it?
Yeah, but I just took microbiome right before I took this class, so.
Okay, so I'm just trying to give you guys as much of this as I can so that when you look at it in the book the second time, it makes more sense. So here's our plasmid. Right, it's our 5,000 base pair, double-stranded DNA. Now we've put in our gene of interest. You know, the protein that, maybe this is insulin. This is something we want to produce. We want to use the E. coli to produce. And we're going to use a transformation process to get this inside of our bacterial cells. Obviously, this is not to scale, right? The plasmid's way smaller than the E. coli. And so, so three things can happen when we're trying to create our clone inside E. coli. This is actually probably the most common one. Most E. coli are not going to take up the plasmid. And the reason being is, you can imagine, to get a piece of DNA inside a cell, we kind of have to put holes in the cell membrane. We actually make the bacteria somewhat leaky so that the DNA can get through. And so the bacteria they're a little bit weak when this happens, and we can't make the holes so big because that would just completely destroy the E. coli. So, okay, so this is the most common outcome, is that no plasmid gets into the cell. The other thing that can happen is we can have plasmid get into the cell, but it was plasmid that did not have the gene of interest. So that's no good. So this cell will be able to grow. Well, and actually, so this cell will be able to grow in the presence of ampicillin. Because remember, our plasmid has a gene that gives the bacteria resistance to an antibiotic. And so this cell will be able to grow in the presence of ampicillin, but it's not going to make our protein of interest. This cell here, if we give the media ampicillin, nothing's going to grow. And so this is an agar plate. It's a plate that has solid food for E. coli on it. And if you plate cells that can grow on it, you get a colony. And the colonies are these circles. And those are just many, many copies of E. coli that have replicated. So on this plate, there will be no bacterial growth because none of the plasmid got into the bacteria. With this outcome, the bacteria will be able to grow and they can create colonies. However, it's not going to be able to make the protein that we want. We need a way to be able to tell that that's the case. The third outcome, and this is the one that we really want, is you have your gene, you have your plasmid, it's got the gene of interest, and it gets into the bacteria. Both of these will be able to grow in the presence of ampicillin, but only this one makes the protein that we want. And the way that you tell that you got one that has the gene of interest is it disrupts. They position this gene in the middle of a protein gene in the E. coli for beta-galactosidase. So this one disrupts an enzyme that, when it's present, the bacteria are blue. And this is the blue-white screening. So it's the presence of our gene of interest disrupts the production of beta-galactosidase. And when beta-galactosidase is present, you get blue colonies. And when it's not present, you get white colonies. And so that's how they select for the 3 different outcomes. That's one of the questions on the homework. And it's a very, it's sort of a seminal advancement in cloning in molecular biology. Okay. So I think those are the things that I thought were going to be most challenging to grasp. I'm hoping that you guys make some progress. I probably should have made some progress towards the virus stuff too, but I guess that might be challenging for Thursday. How many hours are you guys spending on the homework, do you think? I mean, I'm thinking it should take probably a good 3 to 4 hours, right? Because this is the bulk of what we're doing during the week, and the class, a normal class should take about 8 hours a week.
Yeah, I'd say homework would take that long.
Yeah.
Around 4 hours.
Yeah. And I think that's about the right amount of time. I mean, technically we should probably be spending a little bit more time on the class, but I think that that's appropriate for what we're doing. So I guess, what are the challenges right now in terms of time and understanding? None?
So sorry, do you mind repeating that?
Yeah, I'm just trying to figure out what I can do to help you guys get through this. So I want to know what the challenges are.
I think usually if I don't understand something, I end up Googling it or looking it up on like Khan Academy and other YouTube videos just to get clarification. And that usually works.
Okay. Okay. All right. So as long as you can find stuff like that. I think when you are stuck on something, I can also look for videos to insert into the PowerPoint and let the class know when I find them.
Yeah, that'd be helpful because sometimes when I look up something, it's like very specific or like all that comes out are academic articles, and it's hard to sort through and read through a bunch of academic articles trying to understand exactly what's happening.
Yeah, that makes sense. So I can identify things that are at the appropriate level of detail for this class. Because there's things that are definitely more detailed and more specific. And then I think sometimes there are things that I'd like to see more a little bit more detail on. But usually I'm mainly looking for a general sense for this stuff. All right, so yeah, send me emails about stuff like that and I can help. I would say on the homework, sometimes I'd like to see a little bit more detail just because it's supposed to be building up your understanding. But I was really pleased with how everybody did on the exam, that was really nice. And so the next exam will be, I like this format and I'll continue the same format for the next exam, but we also got to pull up our bootstraps and get this project underway too. So I think that's all I have. If you guys don't have specific questions, and we can, I can also on Thursday answer questions about the homework. Try to look ahead if you can on the virus stuff, although I still have to get that up too. So maybe on Thursday, I'll Zoom and we can go over this homework and maybe bounce some ideas about the project off on each other. Sound good?
Yeah, sounds good.
Okay. All right. So if there's no questions, then I'll end the session.
Are our grades posted yet or not yet?
I thought they were up. I posted them quickly, so I'm wondering. And Dustin said he saw it, but but Daniel didn't.
Yeah,
I-
they're on Canvas, and I forget, you either have to go to like assignments or submissions, and there's like a little thing that says, shoot, like view corrections or view notes or something, and so you can see all the feedback you got for it. And
Oh, okay.
But it didn't appear in the grades yet.
I think it did.
It
I
appears
don't
in
remember.
mine.
Oh,
It, Alright.
I'll double check.
well, sometimes it might be, sometimes they don't come up, and I think it's because if I might have uploaded the feedback, but there might not be, if I didn't hit the submit button, it might not appear in the grade column. So I'll go back and look through that.
Okay.
Yeah, because I got on those pretty fast.
Yeah,
Um,
I was impressed with the grade time for that.
I was pretty excited to read them, and I actually, I mean, I'm glad that our class is so small because I couldn't manage that with a bigger class, but I really enjoyed reading all your answers to the questions, and I for me it was probably one of the highlights since we stopped meeting. So all right, yeah, I'll go do that now, and if you don't see it, let me know, send me an email.
Okay.
Okay, so stay healthy, you guys.
Thank you.
Thank
Thank
you.
you.
All right,
Bye.
bye.
Bye.